So if it's all right with you, I thought what I'd do is I'd try to summarize, sort of put all the different ideas we had yesterday and make them seem more normal, like consistent. And the more I thought about it, the big insight, or the big odd thing that we came up with a long time ago, but we're also dancing around yesterday, and Viviane mentioned it, is that the idea that there's two types of models in a cortical column. There's a morphology model, and then there are composite models. And this really confused the heck out of me for a long time, because clearly there are composite models. I mean, you can't argue there aren't composite models. Things are composed of other things, and we can rearrange them. But then there are things like circles, or an egg shape, or an oval that have no components, and yet you recognize them, right? They have a shape, but they don't really have any features. So, it's like I can't point to, oh, this thing is here at this location, this thing at this location. So that was always like a really problem for us. So if we just adopt that idea that there's two types of models.
How does the composite model relate to like the feature model that we talked about?
Same thing.
Same thing. Okay, so
If it's got, the odd thing about, like, in a primary sensory region, there's no subcomponents,
yeah,
but there are things like color or texture, and like if you consider those as components, that they're not related necessarily to morphology, but there's something at a location that can be read at this location. I don't know if that's what you're getting at.
yeah, I guess I'm asking, so if we have a model that's purely morphology, so it only contains information about like the point normals and relative locations, but it has multiple parts, like it's the model of a human with arms and legs. Would that be a composite model or
I don't know.
morphology?
You can tell me. Let's go through this conversation.
Okay.
And then,
Yeah, go ahead.
Yeah, because I feel like we have kind of a approach to this that, but yeah, I definitely want to hear the whole thing, but my initial concern with this is it feels like the morphology models are only at the bottom, kind of. Whereas I feel like we had a way of describing it where morphology models can also be
I
kind of
don't, I'm
compositional.
not sure, I don't think of it that way. I just think, okay, here's a column. And how would a column do both of these models? Now, maybe I didn't think about, like, oh, what would a V4 column? How would it be have a morphology model? I don't know.
Yeah, I think just the way we talked about it before was that there are two models, morphology and feature model, instead of morphology and composite model.
Well, that's the same thing in my mind.
Okay.
You want to call it feature model, that's fine. A feature can be a component, or it can. All right, one is like point normals at locations,
Yeah,
and your other thing is something specific beyond point normal at that location,
yeah,
Yeah,
right? I was
Beyond
calling
the
that
shape,
composite.
yeah.
If you don't want to call, you want to call it feature, fine. But we started using the word composite elsewhere, so I look at, you'll see in a moment here, we think there's like two types of input to layer four. One defines a point normal, and the other defines what is at that point normal. Maybe nothing, maybe it's just a
Yeah.
point normal.
yeah,
Or it could be a coffee, it could be a logo, or it could be red. I mean, in my mind, those are all sort of on a continuum there. I don't make distinctions. If you don't like to use, if you want me to change that to be a feature model, I'll say that. But the feature can be other objects.
yeah, I agree.
Would you like me to call it a feature?
It would be more clear for me.
Okay, sure.
yeah, I don't know. I mean, I don't.
How about I just write it right there on the
slash feature?
Yeah.
Um,
But conceptually, the important idea is to keep the distinction, right? One is
Yeah.
just there is a point normal edge or something. It could be internal edge too, it doesn't have to be just external edge. But there's no other details beyond that. And the other one is, oh, there's some detail at some location that's different. And there's a whole bunch of different details that might be there, features, including whole other objects.
yeah.
You have a concern about this?
No, because I'm also remembering now how we talked about with the kind of direct connections to higher levels of cortex, that would basically be what creates the morphology model in the higher level.
Maybe. I don't know. I wasn't even thinking about that yet.
Okay, yeah, that was at least one possibility we discussed where yeah, with the person, so you kind of would have like a broader, more coarse point normal over a bigger region of space. And then you get this kind of more coarse grained morphology model.
Can we just,
As opposed to like thinking about, so that gives you like the stickman kind of, but then you could also zoom in on an arm and then you would have the morphology model for an arm.
I'm confused about this.
Okay.
Can we put that, can
Sure.
come back to it?
Yeah,
Yeah,
sounds good.
I'm a little bit stuck in my own.
I
level.
remember last year we had like all these pictures of stick figures.
Yeah, and then they
Board.
started dancing, and then there was this like Elvis Presley dance that.
I really want to get to that.
But yeah, sorry.
Can we just, I want to cover what we talked about yesterday, I
Right.
think,
Yeah,
the idea we had yesterday, and then I come back to this. I wrote it in there as hierarchy. That's your cue to tell me about this problem
Okay,
again.
sounds good.
Okay. So in my mind, it was this idea that there's these two different types of models. They're really kind of separate. You can have a morphology model without any features, that would be just like a circle or an egg shape, an oval, or maybe it's a cylinder. I don't know. I don't know what the extent of it. We'll come back to it in a moment because I have questions about the capacity of the morphology model. And then you can have specific versions of that. You have a morphology with specific features. And then it becomes unique. I could have a cylinder with handle or a cylinder without a handle. I could have a cylinder with a logo or a cylinder without the logo. To me, this is like, I know we've talked about this fast, but I've forgotten about it. I came back to it, the clarity in my mind that's driving all the confusion that in the conversation we had yesterday. So before I go on, is there any other thoughts about that before we go on? That there's these two types of models. And then we can work from there, the issues.
So the features would be more like colors and.
The features, I'm going to describe in terms of the anatomy. The features come in to layer 4, and there's a large variety of what they could be. They're basically saying there's something at this location that's not a point normal.
and that's like also the thing that would explain like recognizing the fruit face, because we have the morphology of the face, which is still a relative
Right.
location of eyes and nose and mouth.
Yeah,
But then the features are just different, like it's just different object IDs.
Right,
Like those
right.
The
locations.
sub handle is still
I don't know yet, okay? Where the border is between morphology and features, it's a little hard to say at the moment. I think it's a fuzzy border. Take something like a word. A word is composed of a set of letters, so it's clearly a composite feature-based model, right? It's like these letters in this position order. But once we learn to read, we also we create a morphology model for the word. And I can read a word without looking at the individual letters. I just recognize the shape, and you can fool me. I can drop things out as long as it's got the basic all shape. I'll still see it if I'm reading through. I won't even notice if there's something missing, you know. And you kind of recognize it by shape. So there seems to be like some border. There's some sort of spread between these. It's not necessarily clear. So that's another issue. I want to get back to what the idea from yesterday, okay? See, the idea from yesterday was, and maybe we had this before, I don't remember. So this requires, first of all, requires two sort of separate reference frames. You have to have one that's very unique for the unique objects, and you have one that's less unique that you can use for the morphology object. But they're tied together, and they're not sort of independent. They have to be updated together. It requires two types of input to layer 4. And we do see that in the cortex. There's one would be to determine the point normal, and the other would be representing whatever that feature is at that point, if there is one. And so if you look at the inputs to a cortical column, there's an input to basically that goes on to the dendrites of the distal dendrites of layer 4 cells. And we used to think like, oh, this would be driving, you'd have to drive the activity of these cells would be on the proximal dendrites, but it doesn't look like that. They're on the distal dendrites. But we also see a layer input to lower layer 3, right at the border. And I've always believed, and I don't have, there is evidence for this, but I have to go back and look at it again. There are these cells I was mentioning yesterday, these bipolar cells. The specific ones are called double bouquet. I couldn't remember that yesterday. That seemed to be these cells very skinny, very branchy dendrites that sort of go down one minicolumn. They're controlling a minicolumn of some sort. And so my guess on what's going on here is that this input here is determining the point normal. The point normal is basically the edge orientation of these minicolumns. So these cells, these double bouquet cells, are complex cells. They have a lot of synapses. So the argument here is that the spatial pooler is actually occurring at the level of the double bouquet cells. They are essentially saying, my whole mini column is active, but these guys are doing the spatial pooler operation. And so, that's one piece of it. So that was required to sort of two types of features. Essentially this would be like color and this would be point normal information. Or this would be, from the previous region, this would be called, logo. And this would be like the edge of the coffee cup or something like that. Then we talked a lot about two different reference frames. It's required for two different reference frames. And that was a real challenge. We talked about how do you have two reference frames, both simultaneously updated. We talked about two possible ways of doing that. One was to use phases. That is the same set of cells. At one phase it's sparse, the other phase is dense. We also talked about having multiple sets of cells where some of those are dense, and then below that there are sparse versions of that. I'm going with that one at the moment. Okay. This one's a little bit harder to understand. This one might match. I don't know. It may not be right, but that's the one I decided to go with. Partly because you can now see parallel construction here, which is nice. Doesn't mean it's right, but it's nice. In the lower layer, this would be this is layer 5, and this is layer 6. Say 6A. This is layer 4 here. The input to there's a sensory input to the to right at the bottom of layer 5, right above. It's very analogous to the one that's right here. I don't know, but I'm going to argue that there are double bouquet cells that control the upper regions and double bouquet controls the lower regions. There's literature on this from a long time ago. I haven't looked at it in a long time, but there is some knowledge about the connectivity of these double bouquet cells. So I'm just going with the idea that the same thing is happening down here in some sense, that you have this input here, which is defining your set of grid cells, the classic grid cells, however they get created. So, there might be like in the tank paper, there might be 6 grid cell modules, and each one is sort of there's a cell active in each one as they move around. And then the idea that below them, below the active ones, there's a more sparse representation somehow. We talked about different ways of doing that. One, each little slice, or the random SDR learned, that was another way of doing it. The idea here is you got two sort of representation. You'd find that the standard grid cells, and then there would be these very sparse representations, which would be difficult for most researchers to see. And then the idea here is you have the two models are defined between the black connections here are basically saying, hey, based on this point normal and based on the crude location of some sort, not very sparse location, I could define a model of a morphological model of an object. There's some issues with that, but that was the idea that we came up yesterday. And then there was another set of connections would be between the sparse representation here and the sparse representation here, which would be the model of a unique object that has that morphology. Right? So the same morphology, but now unique. So I can have a lot of unique versions of basic shapes, basic morphology. So that was a new idea yesterday, that you'd have these two types of, our classic model between location and feature. It's really, now there's two of them, and there's two reference frames, and there's two sets of feature type of things. So that was pretty cool, I think.
Yeah, I really like that. I mean, we talked about it before, like having the morphological and the feature model and how that could work in layer 4 and lower level 3, layer 3. But it's a nice idea how that could tie into the layer
Right.
6.
I don't think we had that idea before.
Yeah, I think this connectivity, that's a new cool idea, I think. I don't know what you think, Niels. You weren't there yesterday, so.
In terms of the connectivity, or?
Yeah, that's how they could connect, and that's why it's.
But at the moment, don't ask yourself, does neuroscience look like this? Does this make sense logically?
Yeah, but I mean, yeah, because we were talking before about the two models, and I think that definitely makes a lot of sense. And I think what we were talking about before with the sparseness of the lower level, I think that's particularly important because, for example, even just how we would implement it in Monty, we don't want to just double the number of models we have of everything.
Yeah.
But because the morphological model is going to do a lot, and then you could imagine the kind of more feature model is just going to do a few points stored in it, basically. So you might move quite far before it even predicts anything.
Right. The feature model may have nothing to say. Feature model is something to say when you get to a feature.
Yeah,
And it would nicely fulfill that requirement number 2, that we want to have to relearn, like say, the whole morphology again, just because we have an object with the same
Right.
shape and different features on it.
Right. This is sort of a very speculative idea here. It's theoretically driven, but really speculative. But the good news about it is that we can probably test it. I mean, there's probably literature out there that this makes very specific predictions about the connectivity between double bouquet cells. And I know I have read at least one paper on that in the past.
What would you look for there too?
I'd be looking for this sort of separate connectivity here, right? There's what do those double bouquet cells project to? And what's their distribution in the upper and lower layers? What do they look like? This basically says there's these two separate types of connections. We might typically classify people say, oh, it's between layer 6 and layer 4. That's your classic view, right? Well, this is not, oh, it's a little bit more subtle than that. It's like these cells are different than these cells, and these cells are different than this cells, and there should be connectivity like this. That basically is a new idea, and I've never searched for literature on that specifically. So we might see some papers which just say that this is impossible. It just doesn't look like that. Or people have measured the the receptive field. The response properties of these cells, and there's some literature there. It's not a big, but there's literature there. And so I'm just saying that this is wild. If you're a neuroscientist, you say, oh, this is completely speculative. It is speculative, but it's theoretically derived and not used to that. So there's a lot. If we believe this is true, we keep. I would have never thought this idea there's just two models. 5 years ago, I was just crazy, but we kind of worked our way into that, believing that. And if that's true, then this is a mechanism that seems there aren't too many mechanisms that would work, and this is the only one we've come up with. It's the only one I can think of. So then there should be literature out there that supports it or invalidates it. So I'm just saying it's testable to some extent. It's not like it's not one of these things you say, oh yeah, it could work anyway. No, this is very specific predictions about these connectivity. Even these cells, the idea that you have grid cells, but then you have other cells that are sparse versions of grid cells. That in the grid cell literature, we might find that too. I mean, there's all kinds of grittiness cells, but this is a very sort of specific prediction that we're looking for. This very sparse connections of cells. These cells on a particular object would always become active in the same location on the object.
basically a place cell.
Yeah, well, interesting thing about that one. I don't know. Maybe.
And so then we don't need like a whole.
Are place
hardware.
cells more sparse than grid cells?
I don't know if like by definition, but just as in like,
I
if
think
you,
they are.
if you only need to encode like a few locations, if you don't need to densely encode
Yeah.
space, then it makes more sense to almost like use the grid cell encoding of space and get the path integration and everything.
Right.
And then you just have these additional cells that encode like, okay, there is a unique feature here or something.
Well, I think the idea that all you said, these look like place cells.
Yeah.
Yeah, did you have any more thoughts in general on how we could get these sparse encodings of location?
Well, we talked about the two methods yesterday.
Yeah.
I have no more thoughts about
Okay,
that.
all right.
In some sense, if we could prove that there is a sparse representation like this, then I could say, well, exactly how is it derived isn't that important. Just like exactly how grid cells are derived isn't that important. It's useful to know and just explore, but there's holes in the theory, and it'd be more important to know that these cells exist than exactly how they're derived.
Yeah.
But maybe I didn't think about that further. I was so we put so much crap on the board yesterday. I was all over the place and I couldn't remember things. And so I'm just kind of clarifying what we said yesterday, I think.
Yeah.
And this was the big insight at the end of the day, was that these two different models could exist simultaneously with a denser and sparser thing. And we had to come up with a way that when grid cells are updated, the proper sparse version of the grid cells has to do path integration too. I found it more comfortable just thinking about the 2 cells per layer type of thing, but maybe the other method you said, which is you could pick random, would work too, but you'd have to learn it somehow,
Yeah.
That has always been a challenge. How do you update a sparse SDR for a location? But if I thought of this as many many different grid cell modules, and within them the cells moving based on path integration, then it would work. I could spend more time thinking about that. It would be fun to say, oh, can we come up with a mechanism? How would it play out with those phase distributions and so on? But anyway, I didn't think at the moment. To me, this was just like
Yeah, and then just kind of then playing out kind of like the examples we were talking about yesterday, like the soda can versus the mug. So yeah, because I guess just this whole thing about like something like a handle, which is kind of morphological, how we kind of factor that in. Because like one of the first examples we talked about, it was kind of like, oh, there's a cylindrical kind of morphology object, and then it kind of wasn't clear, are we like transitioning now away from a morphology model when we are then on the handle, or is it just like,
this gets back to the question that came up earlier. I don't
Yeah.
know, right? There seems to be well, let me just bring up the first issue I had here, which is the capacity of the morphology model. How many things can you represent
yeah,
in the capacity of them? And in this scheme, as I've shown it here, it seems too limited. If you think about, if you think of the upper layers, every mini column is like they typically classify, oh, these mini columns represent edges that, orientation edges. And so you may have literally, they may only be representing 8 or 10 different orientations. As you move through the columns, they cycle around. And if you take, there's evidence that that's not right, but if you accept that, then there's very few. Okay, I can say, okay, I have these point normals at this 8 different locations. But how many different locations could I use? And if I just use this sort of one level grid cell for thing, well, there's not that many. If I take the tank paper and there's 6 grid cell modules, but they're all going in sync, then there's like maybe 30 different points on that grid. So I don't know how many of there are, maybe 100, doesn't really matter. Basically, I only have 100 different locations that I can really represent by that. So if I just took the pure dense grid cell model and the pure dense, well, I don't think the upper layers is the problem. The problem is the lower layers. But I just took grid cells and I say, oh, how many different locations can I have? It's pretty limited. So I can't really learn too many morphological models if I stick to the dense grid cell.
Yeah, well, what could be the way to do it is, so if we think of these grid cells implementing transition models, that's like a term we talked about like 2 years ago, I think. So basically saying the grid cells, so how you normally think of grid cells as encoding 2D planes.
Right.
But then let's say in the neocortex, we can have grid cell modules that can encode like 3D shapes, so like a cylinder, and they encode that if you go 360 degrees around the cylinder, you're back at the original location and stuff like that. That would mean we would need custom grid cell modules for the different basic shapes. So we wouldn't want to have.
Why would we need custom grid cell modules for each basic?
Because they need to know like the basic properties of geometrical objects. So you would have one like for a cylinder that knows that you go around it. You would have one for a sphere, and like there are not that many basic shapes. So you know the path integration properties on these 3-dimensional objects. But then I think it would work because, I mean, this is like high school art knowledge, so don't quote me on that. But if we say the cup is like, if you draw things, you often compose them into like these basic shapes, and then a cup would basically be a cylinder plus like a half sphere, like or a torus.
Yeah.
So we would have one grid cell module that models how you would move along a cylinder, and on the handle you would use like the torus grid cell module to know how you would move through this kind of 3D space.
So I wouldn't want to, wouldn't want to have multiple grid cell modules for this, but could the same grid cell module, what you're suggesting, the grid cell aren't just encoding your location, they're also they're somehow tuned to the action.
Because they would need to know some special path integrating properties that you only have on those shapes. Like in 2D, on a 2D plane, you wouldn't have the fact that if you exit the 2D plane on one side, you
Yeah.
enter it on the other side again.
But
Well,
I
it
just
does do
pointed,
that, but
just
yeah.
put it, I guess is like, I guess you could model it, all of these with like a 3D, just like a 3D coordinate grid cell module. But then it would be a separate thing to know like what actions are valid on a particular object.
Right.
So like you would still get path integration if you just had the cylinder model
Yeah,
embedded
so-
in a generic 3D.
By the way.
Yeah, I guess the alternative would be like what, I think that's what you're saying, that to have just a 3D grid cell module
Yeah.
that models 3D space. And I guess I'm just saying, maybe it's not a
But
maybe not like that, but
issue,
instead it's like,
I think this issue you come up with, it also exists in 2D space. You know, a rat will follow these mazes, and they go up one arm, and they go down another arm, and they come around, and they come back to the beginning again. They show that they model those are like separate spaces, but whatever, but the point is, they don't really know they're back at the same spot unless they observe the same feature.
oh,
Imagine
they
that,
don't?
yeah,
I thought
imagine-
that's like, one of the key things.
but
of
only
path
path integration only works within a. It
Or
doesn't
just that
work,
it's noisy.
it doesn't really work long distances like that.
Oh, okay, yeah,
Imagine you're walking in a big house. 100 rooms, and you're wandering around. And you went through one room and you come back. You might be surprised, oh, I was in this room already. You you didn't say, oh, I'm expecting to see that particular bedroom again, because you really kind of lose track, you don't really. So you're like, you kind of build your map and, within some local area, the path integration works. But as you start going further and further afield, you really don't, it doesn't work too well. And you could come back and you might be surprised you're in the same room, or you might be in a different room. You wouldn't be certain where you are. At
yeah,
that
but
point.
maybe that's more of a scale issue, that they don't work at that large of a
I'm just
scale. It's
saying
like
that
with 3D objects,
I-
you usually don't have that.
My intuition in from the literature I would have is that these ideas that you come back to the same place again, that the grid cells wouldn't know that necessarily in this sort of three-dimensional circling.
Well,
object.
ideally, if you're receiving, like updated signals, when you get back to the same location, the same grid cells are active. But it's just, if you're relying entirely on like path integration, on dead reckoning, then that's not guaranteed. But like, if you're going around the mug or the house or whatever, and you're getting updated information about which room you're in.
Well, this really has to do with the fact of what do we do about edge wraparound, right? That's the only issue here, right? The only issue is like you run up the one edge, you have to know you're coming back to the same edge on the other side, where you don't seem to get that in a two-dimensional plane, even though I'm arguing it does happen because you don't really know where you are. So this is like, okay, yes, does the grid cells know in advance that they're coming back to the same location and through path integration? My intuition says they wouldn't know that. You'd have to learn that. It's too much to ask them. In fact, all the evidence we have for grid cells so far is that they really do best in 2 dimensions. They don't really work well in 3 dimensions. Remember the bat papers, we read those?
Yeah, but that's why I'm suggesting that we don't have this approach where the grid cells are just general 3D modeling units.
Right.
But instead we have this approach where it's more like a 2D plane with the added knowledge
Right.
that we. end
So
up
I would
back
say
with.
my intuition would be, it's a 2D plane without that knowledge that you've wrapped around and that you discover that you've wrapped around. And you say, oh, I'm back to where I am again.
Like, yeah, but I guess so if we, for example, have a cube.
Yeah.
The grid cell module would model more like a space like this. Like how you fold a cube out of paper,
Yeah,
and then.
but that's
you would
only
know
one
that,
folding.
like, this way.
That's another,
like
it's
that.
only one folding. There's other foldings where, you might wrap around the square on the bottom there, you know what I'm saying? What happens if I go this way? This one has to go someplace too, and this one has to go someplace too.
Yeah, so that would be the extra knowledge that the grid cell would have to encode, basically.
Okay.
So this is, that's kind of what, for example, like Philippe George's, where it kind of explored, where he took essentially like HDM type stuff and then extended it to like just learning sequences where you learn every transition possible, like you visit every edge. And so that's like one way to learn it, but then you're not getting the benefit of grid cells, which is like out of the box, kind of generalized to novel
Yeah.
paths.
But I wouldn't want them to learn this like for discrete transitions, but like continuous space. And basically what I'm saying is there are not that many basic objects. So we wouldn't have to learn this for like all the possible
Yeah.
objects in the world. We would just learn this for like a sphere. a
Well,
cube, a
so
cylinder,
I
and-
think that the system has to make no assumptions about what the basic shapes and objects it's gonna learn are. And it just has to do with anything. I think this is, I don't think this is the way it's gonna go. There's too many weirdnesses about this. And grid cells don't seem to do this, and we also have to match it onto the anatomy, right? I mean, I don't have.
But like,
Um,
didn't you also say that they don't work very well in 3D space?
Right, they don't. It's almost like you're doing 2D and then 2D and then 2D and somehow you patch them together.
Yeah, that's kind of what I thought that.
Well, but no, but it's like take a die, right? Like a die from playing games. Most people have no idea what on adjacent surface, what the number will be. But there's a pattern to it. They're all the same. And no one learns it. They don't learn to do what you just said. It's almost like you see a face and then I see another face and I see another face.
But I think it might be a different issue there, because you know with the dice, like how you move around it and where you get like more, if you just look at the morphology, not at the features, you know how you would move around the dice, and then if you go around 4 edges, you're back in the original space. I think the issue you're describing is that you don't learn the features on the different edges and which edge they're on, but that might be because it's a symmetrical object and you don't really know how to anchor them on the dice. So if you have like a
But if I have a unique representation in the morphology model for every location on that, why? I mean, path integration should tell me what's on the next face.
But maybe you don't have a unique representation here because it's symmetrical, so you don't know which.
Well, but you
face-
just argued there that I did with the
No, but is it like
unfolded
even
cube.
the features don't tell you the orientation of the dice, like you
Right.
need to look at 2 faces to know the orientation of the dice.
Because
And
if you have
even then,
something like,
even then, you won't know what's on the other faces. You just don't learn it. You just don't learn the transitions from the face of a cube.
but if you have like the coffee cup, which is not as symmetrical, when I started the Numenta logo, I know like I could like take a blank coffee cup and put them back on there like they are on this one. Like I would know how the logos are placed on this cup.
Yeah, I didn't follow that argument, but,
Like I would be able to draw the same feature structure on this cup from memory, because it is
Yeah.
not symmetrical. I know the
right,
handle is here and
right. But,
there's top and bottom of the cup.
again, I've always been really surprised how, again, like you can't really, if I asked you to draw the cup, you could draw it like that, but if I asked you to draw it at some angle like this with the letters, it's really hard to do. You just can't recall that. It's like you don't have a map of what it looks like when it's at an angle like this. You only have a map from planar views. And this seems to be an observation. I'm gonna go back. I still think there's a capacity issue here. Even if you're right, Viviane, that there's different grid cell modules for different shapes, which I'm loath to accept the moment. I don't know how that gets represented up here, right? I just don't know how it gets, where are they? How many are there? Who decides what they are? The world could be composed of, one world could be composed of one set of shapes, and another world could be composed of another set of shapes, and who knows?
Yeah, one kind of related thing that we discussed once was like kind of having almost like a view sphere where it's like grid cells on like generic 2D sphere surface, and then you're kind of always like looking in and projecting onto that. So it's kind of like this, but it just means that you don't need different ones for different objects.
Yeah.
And that again would, maybe kind of fit with the dice. Like if you just can't really orient it, then your view sphere is just going to be a bit of a mess in terms of like which specific feature is on which face and stuff. But I remember there were some limitations we felt with that. So
I would much prefer that we make no assumptions about the basic shapes of the world, and the system learns them, whatever they are, and it's going to have some capacity limit, and it'll learn whatever it can from the statistics of the world.
Yeah, I mean, we might, I'm not saying you were born with these, you might still learn them, but then it's like, it takes, you don't really learn new ones after a certain point, or it takes really.
Well, I
long to
guess the
learn
one
more.
question is, can I do this with the same set of cells that learn all these different morphologies up to some limit? And I think what you're arguing is we have different sets of cells with different morphologies, and that's the part I have trouble believing. It seems like you have one set of cells that are going to learn some morphologies or some limit to them, but there's no a priori assumption about how many and what the different shapes are.
Yeah, I mean, I think you could reuse a lot of the cells. You would just need some kind of modulating cells that tell you to like restart at one point, like once you go a
Yeah,
certain distance.
But why do we go that far? Yeah, we got a system right now that you could say, okay, the problem with this one is limited, but in theory, it could learn any morphology. It's just limited. So to me, the issue is capacity, not like differentiation of shapes, morphologies. It's really a capacity issue. If I just take grid cells on their face value and there's no evidence that there's different sets of grid cells for 3D or for cylinders or whatever, as far as I'm concerned, as far as I know.
I mean, it's just, I mean, this phenomenon could be explained in different ways, but just one thing is that it's very difficult for you to learn a totally new shape. Like if you have like a cloth that's wrinkled up and let's say that's a static object now, it's like a weird abstract art statue in the
The
park.
problem is it's a complex shape, or is the problem it's a novel shape?
It's a novel shape.
Okay, so a folded up piece of cloth is a very complex shape.
Well, yeah, but a simple shape, I feel like there's no simple shape that we don't already have.
Oh, I could come up
models
with, I
of.
could come up with one, It's interesting, I always find it interesting that egg shape is a shape we know, but if I was never exposed to eggs, I wouldn't probably.
Well, the
No.
head is kind of egg-shaped, I guess.
Do you know the Greeble objects? This is like.
Yeah,
This is like used in some psychophysical experiments, but
Yeah.
it's kind of like these novel 3D objects that people have to learn.
Yeah.
I mean, even a
They
two-dimensional
can,
object.
or they have trouble
Yeah, they
with it.
can. I mean, they
Okay.
definitely learn better than machines.
But they all come from generic objects, primitives.
So-
Yeah.
So now I just drew a two-dimensional shape, right? I don't know that, I don't recognize that.
Yeah,
But I think if I studied it for a little bit, I would recognize it. It would be all of a sudden, I can instantly grab, after I've seen it for a few times, I practice it, it'll become second nature to me. Maybe this is the way, some face mask look or a bicycle seat or something, I don't know. My point is, I think it's easy to come up with novel shapes that we don't necessarily already have some sort of memory for. I gave the example earlier about words, right? You kind of, a lot of words, you just recognize them by their shape. And I think it's well documented, but I have to check on that again, but it's clear to me, I do that, I don't, that's why. And so those are new shapes that you weren't born with, they're just the shape of a word. And as long as you got pretty close to that shape, you'll recognize it as that word. So I don't think there's some preordained amount of these things. It feels to me like the morphological objects are being continually learned, it's like everything else. And there's a limited capacity, but my problem here is that if I just do with the diagram I did right here, the capacity is way too limited.
yeah.
It doesn't seem right to me.
But just to be clear, like, that's not really a new issue, is it?
Well, if I, this mapping between this layer and this layer, that's new today or yesterday.
Yeah, but the fact that grid cell modules aren't really sparse enough for.
No, that's not a,
reference range.
right, well, most people don't think of them as sort of representing unique space.
Yeah.
They just don't think about the idea of a unique location.
I
That's.
thought it wasn't that Elif Feet, at least that was like, she had some
She's done
paper in
something about
2008. I thought that was kind of what inspired Mensa's work, wasn't? Where
Well,
you get like that was at least, okay,
that was
sorry,
why
not-
we have lots of grid cell modules.
yeah, and then it gives a single unique location.
Right, and so we've kind of adopted that here again,
Yeah,
by saying there's these extra cells below. It's just, we adopted that idea, we couldn't get the map to the anatomy correctly in any way. And so this is a variation on that could be mapped to the anatomy. Right?
Because there are not enough grid cell modules.
Right, as in the tank paper, there's 6 grid cell modules. And they all seem to be moving together. They're not mapping independently. So really, even though you have 6, it's really like 1. And then where are all the others? I need 20 of those, right? And they all have to anchor differently. And I couldn't find any evidence for that. So this is better because at least here they're very sparse. And you wouldn't expect, you wouldn't find grid cells, to be more like place cells, like you said, you know, you wouldn't find, they don't look like, these lowest green cells don't look like grid cells. If you probe them, they just don't look like grid cells. No.
Maybe just for us to get background, you could go through what we talked about yesterday, why these are sparser again,
Why they're sparser?
or like how they would get
How they
sparser.
would get
Yeah,
sparser. Sure, I don't remember.
But and
Because
just
I
clarify,
also-
the black ones would look like grid cells.
The black ones look like grid cells.
Yeah,
And then you were here for the tank paper, we showed the little 6
Yeah, yeah.
modules, right? And then remember, we talked about like, there were some missing ones, every once in a while there was a missing
right.
one.
And also how you could like scan above or below.
Right, and so, you know, perhaps the missing ones, you might be capturing some sparsity, just starting to get some sparsity below some level. So one idea was that, you have a grid cell module, one of the 6. Here's a cell that acts like a grid cell. And underneath it are a bunch of other cells that are much sparser. Basically, they're like a mini column, and if there were like, imagine he had like 6 grid cell modules, and there was an active cell in each one.
Yeah,
One possibility was you go down a little bit, and now maybe there's another layer of here that's very similar. But the difference here is only, let's say, 2 of these modules are active. Like there's 2 cells, the others are not active. So now this is a, you know, 6 choose 2 is 15. So there's 15 combinations of ways you could pick 2 cells. And that you might have like, you know, if you just keep going down, there could be like, you can think of these mini columns here, there could be like a
Yeah.
whole bunch of these layers. If I have, you know, 15 of these layers, and each one I'm picking just 2 out of the 6, so each little layer acts like a grid cell module with only 2 cells active. Then you've got, it would be like, well, this 15, it would be 15 to the 15th number of possible combinations of that. But each one of these layers, these cells would move around just like these cells move around. They're not anchoring would mean like which of the individual cells, which of the individual 6 modules get an active cell. That would be the anchoring process. So each of the 15 layers could anchor differently. That's a little bit wonky. I have to admit, that's kind of like, oh, well, that's kind of a weird idea. How would that work? But at least it builds on the idea that you might have sort of a mini column structure down here, which we do see. And you're picking a sparsity, but actually when you do the path integration, it's not like I'm randomly picking in each one. I'm actually, this active cell is going to move, and this active cell is going to move.
Yeah.
So that was one idea. This, if it worked, this solved all the requirements. Viviane had another idea, which I never really understood. Sorry, Viviane. Which is somehow you have to start with the same basic premise. And then she thought like, oh yeah, but I could just consider this all big, and I could fix some sort of random sparseness down here. Something like that. And I didn't see how that worked because it didn't do path integration on these guys, but maybe you
but
can
I was
explain it.
basically saying, you originally pick a random set of SDRs that are active
Right,
when
that's how-
a specific state is, when the grid cell module is in a specific state. You associate that SDR with that state of the grid cell module. So then the grid cells still do path integration. So when they are back in that state, the same SDR is being
Right, this is the idea is that you don't really wrap around, you just have to remember, you'll have to recognize them back in the same spot and then says the same thing.
Yeah.
So they don't really do path integration in the sense like, well, they just keep randomly changing until you come back to the same spot.
Yeah, but then one thing is, we use, I don't know how it would work with, I think it also works with what you just explained, but that we have a different random SDR depending on the object that we're on, like that the location is specific to the object.
Well, that happens here too.
Yeah,
It would be easier to do it here,
yeah,
if you just pick a random one. I just think the path integration component of this example is wonky. You're not really doing path
I think what
integration.
you're arguing is the path integration still happens in the.
yeah,
higher level.
exactly.
But then I guess the only thing is the capacity issue not still a problem because now you need to have, like, the grid cells that are active need to be sufficiently unique to activate the correct SDR down there.
Yeah,
Well, no,
basically.
I think what you're saying is that this has to be activated, the correct one has to come from up above.
Yeah, so in the beginning, if we don't know which object we're on, it would activate a union of SDRs, and then that gets narrowed down.
Right, it sort of throws away the whole path integration idea, it just doesn't.
Yeah, path integration would still be done by the grid cell.
Yeah, but down here you have no path integration. Down here, it's
Yeah.
just random patterns associated with the specific
Yeah, but
object.
implicitly by learning an association with the.
Right, it's a lot of learning.
Yeah,
That's right. Every point has to be learned. Every single point in that unique representation has to be learned. And so that to me is not really doing path integration. It's like a poor man's path integration. It's like, okay, well, I'll just remember every location in the world. And when I come back to the same location, I'll remember it's the same one. But I don't even, but it means if I move in some direction, and if I don't have this, if I pick a random thing here and I have a movement vector, how would I know what the next random thing is supposed to be if I don't have this feedback from above? I can't predict the next proper location unless I have somebody, I mean, I don't think it would work.
yeah, I think now that I see it again, that your proposal is probably better. I just think that problem we would also have with your proposal, that we need the object information to know which sparse.
Well, I think you would need it to anchor. But once you've anchored, path integration works without any further ado. It would just say, once I've anchored, imagine I have 20 of these or 15 of these little one layer grid cells, but these are sparser. Once I pick which of these cells in each of these, which of the 2 of the 6 are active in each layer, then path integration works. I can just, if these 2 dots will move around, and these 2 dots will move around, and these 2 dots will move around, and nobody has to, I will accurately predict to the next location, even if I've never been there. I would always get to the same SDR.
Yeah, so
Nice.
basically it would keep the 2 that are active in each layer within that square field, and then just move.
Well,
So it wouldn't change
you're right, the
which-
thing as you move, the active cell changes, right? The active cell changes.
Yeah,
And it could move completely, even within this whole 6 thing here, it could move over to the next one here and so on, right? These are artificial boundaries. Yeah, you'd have basically I would pick
Yeah.
2 cells. It would essentially say,
and that way you don't always need the top-down
you
feedback.
don't ever need it.
Yeah,
It just needs to
that's
anchor.
nice.
You just need it to anchor.
Yeah, so in the very beginning, it would still be like less sparse, more like a union of possible.
I don't know what the very
objects.
beginning would be, but you need to anchor,
Yeah,
right? If you did, before you anchor, I don't know what happens, but,
yeah, that's nice.
right, you need to anchor.
Yeah,
Nice shape there.
yeah, sorry, I'm trying to draw at an angle,
So
so.
we need to take like a 3D drawing course at some point.
You gotta practice drawing on 3D. I wanna go fix mine, make them look,
Yeah.
you know.
Last year
But,
it was drawing forks, this year it's drawing cubes.
So I just wanted to mention, because, yeah, I feel like this reminded me a lot of, what I know you've talked about a lot, or I think kind of, similar thing before, where you kind of have the grid cells, and then the columns, the mini columns are the movement vector cells.
That's
Um,
maybe.
but just,
I don't know,
I
that
don't know.
doesn't fit into this scheme, I don't think.
Well, what I was just wondering was, like, if we need these potentially enable the general 2D grid cells.
The top layer is like the general 2D grid.
There's the general 2D grid cells. These are 1D vector cells. But I guess these could have 2 properties. One is, like, there's a lot of them, so they could be kind of sparsely active. And then they could do path integration, at least over local coordinates potentially. Like over small distances.
Why wouldn't they just work like any other grid cell?
Well, what we were talking about with, like, they only kind of work in 1D.
Oh, I'm giving up on that one right now.
Okay,
By the way, that thing I talked about, Minicolumns, you get of the cells that are all sort of in different phases.
yeah.
Other people have argued that actually there are different dendrites in different phases.
Oh,
So
well,
that you don't have to have different cells in different phases. So, it's confusing.
okay,
So in today's conversation, I've abandoned that idea, so I'm gonna just put it off to the side. And so I'm not trying to accommodate that here.
Okay, I guess I was just thinking, I don't know if this helps in that, like, somehow, the active grid cells here are kind of help re-anchor, these to a specific representation that was kind of learned. And then we do kind of path integration over short distances here.
Well, no,
But
but
then-
this is a different scenario. That path integration like this was my phase transition.
Yeah,
That's not what we're talking about here. When I move, this cell would stop becoming active and this cell would become active and then this cell becomes, just like up here.
yeah.
So in this example, this proposal, movements are just like grid cells.
Sure.
They move horizontally in their plane, if you will.
Vertically, don't they? The
No,
phase is
no.
vertical.
Well, we're not doing phase anymore. Remember,
But I'm trying to, okay,
in
I was just
the
wondering
phase
whether-
example, all the cells are active at the same time, and then which one, the phase shifts up and down.
yeah,
So it's not.
yeah, so I'm trying to go back to that.
Okay, well, maybe we can incorporate the two. That would be really nice.
I was just wondering if that helps in terms of, like, this is a more sparse code. And then, so like potentially has what we need to kind of sparsely reactivate or like uniquely encode a location. It can at least do some path integration, but maybe it's more limited over local spaces, but then maybe this is what helps us kind of get into the right space, if that makes sense. Like you always want to be, if this code is going to be meaningful, when you're at a particular location or an object, you need to kind of reinstate the particular phases that are active.
Right, if I think about cells having phase as a derivative of the grid cells, then I would need to anchor the phase.
Yeah,
The anchor would be anchoring the phase, which then reflects itself in which grid cells are active.
yeah,
Remember, those phase cells are not grid cells, or
Right.
the predecessor to the grid cells. Just bear that in mind.
Yeah.
Just to clarify, in this drawing, in the top part, there would be a cell active in each of the 6.
That's what they
Squares,
observe.
right?
That's what Tank sees. That's what grid cell people see, right?
Yeah,
They see, what they, just remember, imagine you don't see a single cell active, you see a single cell most active, and then in the direction of movement, you see like a little bit less active, and a little bit further, it's like it's a phase procession, remember, like this fires and this fires and this fires and this fires and this fires. So you'd have a series of cells here that are, on each phase, they go bing, 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 bing. And the center of it would be the middle cell, right? That's why the grid cell location they always show is fuzzy, because anyway, it's complicated, right? But
yeah, so I guess the general, I'm not suggesting this solves all the problems, but just if it's somehow that like this brings the like higher D path integration,
higher
Which-
density,
higher dimension,
higher.
like, because this is only
the
in.
farce
in
one?
The top, the grid
Okay.
cell, the true
I'm
grid
not sure
cell.
why you say higher dimension, but
Because it's more than one dimension. We didn't agree if it was 2 or 3.
okay. Now you're conflating the whole idea that just grid cells represent 3D. That's
Well, I'm just trying to be open to, I guess, that possibility, but then, and then this is like local path integration. Which, plus sparse, location code.
Whatever, don't lose it.
So yeah, something where like, okay, so for example, at a particular location A, kind of these ones are active, and then you move some distance, and then another sparse code is active.
But you're moving up, but that wouldn't be the case in terms of the grid cells. The grid cells would be the next one over, the next mini column over to become active.
That's a good point.
Yeah, again, remember the actives, I think we should. Here's what I keep wanting,
But okay, but yeah, I see what you're saying. If it's the same exact structure here where it's like the double bouquet cells, but if it's not like the double bouquet cells, then they might be kind of different. This
Right,
one's in that plane and this one's in this
so
plane.
there's a whole series of questions about how grid cells get derived. And it's, as you know, it's a very complex topic. And so what I'd like to do is compartmentalize our issues compared to those issues. So my compartmentalization is saying we have a dense SDR and a sparse SDR, right?
Yeah,
And they both do path integration, not unlike the random thing. It's just like even the sparse one moves. What I propose here is a variation of what we had in our paper there. Right? It's a variation of that, but one where I can say maybe it's compatible with the observations because we don't see lots of these grid cell modules down here that are anchoring different. But this thing would make it work like that could fit the biology. It's the only way I can imagine how it fits the biology, but at least give me a way to fit the biology to say, yes, I have a unique SDR and a less unique SDR, a less unique representation location. So I'm just happy with that. I don't need to worry at the moment too much exactly how it comes about
okay,
because I just have a possibility.
okay. I guess just to say, I guess one last thing, I guess the only concern was like, this needs all the hardware to actually enable the grid cells. I thought, and
It does.
it was this idea, which, yeah, I
Right.
feel like you discussed before is more like, this is the hardware for the grid cells, but it can be reused, like it basically does more than just enable grid cells, like it also gives you
What you would
a
then
substrate
what
for
then you'd see is
the sparse location.
right. Then you'd see as you go down through the core, you'd see a bunch of cells that all look like the same grid cell, but at different phases. I'm not
But-
sure
but
that's
would-
observed.
Would they, because this is the grid cell, this is the vector cell, so they're different types of cells, so it's not like this
Right,
has the same
right.
receptive field as these.
The problem with those phase cells, someone else has to read them out. Well, all right, look, imagine you're right, imagine these are all active at different phases,
Yeah,
right? Well, they could be read out up here because maybe all these activities coming up here, and these guys detect the set that are in phase.
yeah, the
Right.
specific location.
Right, so that could work. The problem I see with that is, then you would expect to see, all right, they wouldn't look like grid cells, would they? They would look like just, they would look like, they would look like just, cells that are going in a particular direction. They're motion sensitive cells, right?
Yeah, going up or
Right,
down.
right, and that's actually not going up and down, but meaning every cell in the mini column would all be like moving in some direction, with
Yeah.
different phases. And every cell in the next moving column, mini column, well,
Yeah, sorry, the active cell would move up or down, but yeah, the direction that they're
not the
sensitive
active
to
cell,
would be different.
in phase cell would be moving up.
Yeah, okay,
They're all active.
okay, so this is when it's the.
So interesting, if you look at go back to.
Because sorry, when we talk about phase, sometimes I think we're talking about, if you imagine this as a frequency band, then you could be the phase within that, or it's phase relative to
What I was talking about is that.
like a background.
The idea was that you have a set of cells, they're all firing at the same frequency. They're all, basically the frequency is based on the velocity of movement in a particular direction. So they all seem to be, it's their firing rate represents the movement in that direction. But if you look at them, they'll have different phases over the beta cycle or the theta cycle, right? So they all look like they're all firing together, but their spikes are offset. 360
But
degrees.
couldn't you also just have a sparse code where one cell is active and that cell, like it's essentially a 1D grid cell where, or
No, because, well, I have to go back to the grid cell theory.
but then like as you move up through this,
That's
you
not,
would-
what-
yeah, if you're moving in this direction, then this cell would be active and then this cell and then this
but
cell.
no, but not, the mechanism is how to do that. The point of the whole point of the thing is that the grid cells, a grid cell has to look at all these all the time
Yeah,
and pick out which one is in phase with the beta, gamma rate, whatever. What is it? Beta. No, same to me.
okay, well then, I mean, yeah, if we can't do this, then this is a problem because then this isn't a sparse code and then we need some sort of like temporal.
Right.
Temporal.
Well, it is, well,
layout.
the interesting thing is, if all these cells are active, like I just said, and you're taking all those cells and feeding them up here, in essence, it is a sparse code because it's very sparse. One of these cells is saying, I'm only going to respond to everyone who's in phase. Everyone's in the same phase, part of the phase cycle. And therefore, it looks like a sparse code. I mean.
Yeah, if we have like a temporal code, yeah.
Right, well,
read out
that was the speculation that whoever wrote, that, you need something like this to get grid cells to work. And it's so, there's a lot of questions that are streaming through my head right now, but if you look classically, going back to Hubel and Wiesel, they see cells they characterize the cells in the lower mini columns as being directional sensitive. And there are some papers that show that directional sensitivity is different than the directional sensitivity they see up here. So we can assume that's like a movement thing. So these kind of cells, all these cells are firing in sort of the same direction, is what Hubel and Wiesel observed. I don't know if they ever looked at the phase. They probably didn't. But they did observe all the cells in the mini column down here seeming to be, the ones they report on, right? Remember, if all these cells, they don't report on the ones that don't fire or fire very rarely. They only report to the ones that fire constantly. There's a whole bunch of other cells that are not firing constantly. So, it's only maybe less than half the cells have these properties, but that's what they report. So there are some set of cells down here that look like this. They all look like they all velocity sensitive to a particular direction. They didn't report on a phase shift, but if there was a phase shift, that would fit somehow into the grid cell theories of how grid cells are. And then you could take these cells, when I look at this projection up here, if I just look at the population activity, it doesn't look like a grid cell at all. It looks like, it looks like a bunch of cells like this. But if I were to recognize which ones are in phase at any point in time, it would be very sparse. It would be like, in some sense, in some sense, these are almost like reading out a location. So imagine I'm one of these cells here, and I have a dendrite, and coming in on that dendrite is all these different, axons from here. Well, they're nearby, coming nearby. And at any point, and then I fire, and I want to learn to recognize the pattern. Well, I would learn the ones that are the ones that are actually firing in phase, and the other ones would look like they're out of phase. So I would say, oh, form synapses to these. And so in some sense, this dendrite of this layer 4 cell is acting like a grid cell. It's saying, oh, I recognize a specific location. This is a specific location because these cells are all peaking at the same time, and therefore that's the only ones I'm going to pay attention to.
Yeah, or a place though.
It's like it would be like.
But yeah, basically it responds to a specific location.
It responds to a specific location, although these cells themselves don't look like they're responding to any specific location.
Yeah,
they don't.
I guess the only thing is just then I feel like, but this is still important. This layer is still important because. Yeah, these can't do path integration over larger spaces very well because each one is only integrating in one dimension.
Well, they do work. If they were constantly active, they would do path integration because you can think of it as the quote, the active cell is the one that's in phase with the beta cycle, right? So which one is in phase with the beta cycle tells you where you are along some dimension in space.
But that's why I mean it's only local in that because we talked about the example of if you start going in a different direction, or even if you just did something simple, like going in a square, you're not going to necessarily reactivate the same ones. But that's what this can maybe help with.
Right.
So this kind of re-anchors these.
Right.
Periodically.
These have to be path integration.
Either these re-anchor them, or I guess the features that you're observing.
This is a very interesting idea. I mean, I don't know how much time we should spend on today, but it's a really interesting idea that I take the idea of the mini columns representing this, and there are a whole bunch of series of problems here with this. These cells only make sense as you're moving in this direction.
Yeah.
And when you start moving in another direction, these guys don't represent that. In fact, the opposite. If I go in the opposite direction, the phase doesn't shift down here.
Right.
There's another set of cells that represent this direction. So you have to anchor these cells phase-wise and then start tracking the distance. So in some sense, this mini column of cells here represents where you are. The phase represents where you are along some distance in a particular direction, as long as you're moving in that way. Or moving one way, but if you're moving the other way, they just don't represent it. The other guy represents where you are. So it's not, it's like it's a one-dimensional grid cell, but only in one direction. In the other direction, you have to have a different one-dimensional grid cell. I mean, these are really interesting topics. I'm really interested in pursuing this idea. I'm just wondering, have we dealt with the basic issues here well enough? I mean, this is like a detail how you'd implement this. Now, it's a really interesting idea that you would not see these as grid cells. You would see these as just direction-oriented cells. And those all get passed up to here, and these guy recognizes them as unique location codes. I like that idea a lot, actually. That the actual detection of the in-phase cells might occur up here. It doesn't explain then why do I have grid cells down here, because the grid cells have to do it too, right? If there are real grid cells here visible, then I mean, these don't look like grid cells. These look like almost like place cells here, actually. Because this is like this cell will become active at a specific place with a specific feature. These would look like directional sensitive cells. And I still have to be able to generate, why do we have grid cells then? If this is the predecessor to grid cells, these are not grid cells, these are required predecessors to grid cells. Well, we have to generate grid cells, and what's their purpose?
I feel like I mean, I don't know if this would actually work, but it just feels like their purpose would be to kind of, it's like a recurrent connection.
Right, I agree. That's my opinion too. That these are a way like, this is like your temporary memory, like, this is where we are right now, guys. So, you're going to forget where you are. I'm going to tell you where to invoke the right thing. It's sort of like this, some sort of like a holding place. It's not very high resolution, though. That's the interesting thing about it. It's like, it's not very high resolution. But,
And I guess I don't know if you have predictions from L4 going down to the mini columns in L6. If I guess that,
well, that connection our theory is that
would bias it.
you would have to now, what would happen here? You have a cell that's active, which for a specific location, with a specific feature, or a set of cells that represent that, and now you're projecting that down here.
So that because this is going to be a fairly coarse signal telling it which, or like this is kind of predicting which one should be active. I'm just thinking like,
well,
there's going to be a lot that are predicted, so it's almost like the same.
This is the sparse population activity up here.
Yeah.
And this sparse population activity is sufficient to specify exactly which cells down here should be active, or in this case, which cells should be active in the right phase. It would be like it would be like, okay, this pattern says you have to be in the peak of your phase right now, or something like that.
The other thing is it a bit easier to simultaneously encode multiple hypotheses with this?
I don't see how you could, because
you could have different
well,
phases that are all.
But you can't have
like
the two cells. You can't have two cells. Either a cell is in phase with the beta, or it's not. I mean, it's like, I don't see how you could have multiple cells that are.
So you're saying it's at the peak of the beta?
Yeah, the idea is that there's yes, they peak at the same time as the base frequency, and therefore, and the other ones don't, and therefore, anyone's looking at both of them. I suppose you could try to have multiple cells here, basically at the same base frequency, peaking at the same time. But I don't know, it seems like the mechanism would be like, no, we're going to force these guys to be phase distributed. There's going to be a mechanism that forces them to be phase distributed. I mean, these double bouquet cells could do that beautifully. They send an axon down here, and all you need is some sort of delay in, literally the physical delay going down could do it. I mean, it's interesting to know what the axonal propagation properties of these are. Maybe they're slow enough to do that. I don't know how you could do multiple hypotheses here. If you had multiple, well, something would happen. What if I had multiple SDRs here? Let's say I had two active SDRs here. I'm uncertain, I have some a union up here, and I first bring that union down here. Well, it's going to be conflicting data. It's going to say some of these cells will be like hey, I should be in sync with beta, and then this guy says, I should be in sync with beta, who's gonna win? If these two SDRs up here were in different locations, meaning on the morphological object, there are different locations on the morphological object, then you'd have different mini columns that would be like this one SDR would be trying to set one set of mini columns in phase, and the other would be trying to set the other set of mini columns in phase. And then you would have two hypotheses. You'd have two sets of mini columns, one set to represent the phase set to represent one SDR, and the other set to represent the phase of the other set. So you, in some sense, if they weren't overlapping, if they didn't have the exact same location in the morphological object, then you could have a union down here. If they're on the same location on the morphological object, then you have to pick one or the other.
You mean the union would be activated by the layer 4 representation, or?
Imagine I just have
Narrow
two
down.
SDRs.
Because I thought the screen connection is an inhibitory connection, and the layer 4 representation just kind of narrows down the cells.
You're saying you thought that because that's what the neuroscience
Yeah.
says?
Yeah.
Is that true?
I thought
there
that
was
was also in Nomento's papers before, that the layer 4-6 connections. If I remember it right,
I wouldn't think that.
let me see.
That may be true. Remember going back to the Thompson paper? Where she showed,
Yeah, in Thompson, it says strong reciprocal inhibitory connections between layer 4-6 and layer 3-5, 3 and 5.
does that mean it's only
Through
inhibitory?
large basket cells and double bouquet cells.
I don't think, that could be true, and also at the same time.
But yeah, they might be excited.
Right, excited too, because go with the Thompson paper, I remember she showed those the axons rising up here and forming lots and lots of synapses up here.
Yeah, I think
And you're saying those are inhibitory synapses?
so here,
That's not the figure.
Yeah, at least in the Numenta code, it's excitatory. The main inhibition is implied in the composition.
Yeah, that's
of
one
the
here.
Minicolumns.
So like you have these are all axons coming up here, right? And they're forming all these synapses up here.
I can put it on a little big screen.
Yeah.
Yeah, I was confused at first at why the layers underneath the dense grid cells have to also follow the same grid cell.
Do you understand now?
I'm leaning more towards the way.
Well, the other one on the left there.
Yeah, because I was also thinking it's more like mini columns, the way we have the layer 4 and layer 3, the bouquet cells.
Right. The important thing is that, and I had to convince Viviane of this, that path integration has to occur automatically, even for sparse representations of space. It just has to, that's how you have to do it. I mean, you have to, if I'm in some location, on a unique location on an object, and I move, I have to always go to the right, the same one. And you can't learn, it has to just, that's the whole point of path.
Yeah.
Anyway, well, it's interesting idea of combining the phase precession thing on the left there as an alternate way of doing what I was suggesting here, which I thought this was kind of wonky, but it would work.
Yeah.
This is a little bit better.
Because it would still also encode the way the grid cells move from one mini column to another on the top of the dense.
Well, right, it's like this guy here would have to say what mini column to go to,
Yes.
and then
And then it would encode the path integration in the in which cell gets depicted rapidly.
right,
So
although
here.
I don't know how it deterministically set. It's like what if I move from this mini column to this mini column because I'm moving in some direction. Well, I'm moving in some direction. This guy has to move. Yeah, see, there's no longer a correlation between these cells and these mini columns. That's no longer the case. This is not. These are not related to that actually at this point. They're totally separate.
They're not related to the, so when this one moves from here to here.
That the mini column doesn't move from there to there. No, that's not the way it works. I mean, it looks like that's the way he drew it. But would it really be like this? You've got a set of you just have a set of mini columns here. And then you have a layer of cells up here. These are the grid cells, these are the grid cells in here, but these grid cells do not correspond to these minicolumns. They have no correlation at this point in time, because when you move, if I'm moving in one direction, these minicolumns aren't changing. It's just that which cells in phase are changing. Whereas these cells do change. These are moving around on the grid. These are just the in phase cell moves up and down.
So they're not driving this the same way that the bouquet here are driving.
They're driving this, but not in the column basis.
But
the-
why?
I'm just helping Mohammed with something, so I'll be here in a couple minutes. So
Sure.
yeah.
Yeah, I know it's a bit confusing, but think about it this way. I'm moving in some direction. The grid cells, the active grid cells change. But all it means is, but if I'm moving that direction, there's a cell in this column, mini-column represents movement in that direction. The mini-columns is active though throughout the entire movement in that direction. And all I'm doing is moving the in-phase cell up and down. And because it's a velocity controlled oscillator, so it's firing a little bit faster, so all the cells are going a little bit faster, and therefore which one's in phase of the beta cycle changes. So when you move in a particular direction, the phase of there's a couple several mini-columns that are active, but as you move, which mini-columns active doesn't change. It just says, okay, I'm moving in this direction, therefore which of these cells become in phase with the beta changes.
Yeah, that's if you assume that the phase is on the mini column.
Well, that was that proposal. That's what Niels proposed. I mean,
Okay.
I made this proposal. He's trying to bring it back in.
If we don't assume that, then.
But then they don't do anything. Then we're back to this, right? Then we're back to this idea where the mini columns represent locations in a grid cell, and so which mini column becomes active that's this idea.
That's this idea.
That's this idea.
Then this is the one I understand.
Yeah,
I'm still a little hazy on the phases and the.
if this idea is you have a set of grid cells here, and then these mini columns are a predecessor to this, but the mini columns are not tied together. This is a different set of cells. It doesn't tie to mini columns. It just each of these grid cells are detecting patterns, in phase patterns down here, but it's independent of the mini columns.
I was hoping it was as simple as we have a dense layer and we're just trying to incorporate context into them or.
That's what this
pattern
idea
information.
was, right?
Yeah,
That's
I
what this
was
idea
hoping it was as
Yeah.
simple as.
So talking to Ramy here, it's confusing. In this idea we're pursuing, Niels, which I really like a lot. If you have a layer of grid cells, those grid cells are they don't have any correspondence to the mini columns beneath them. Like a grid cell that's active doesn't mean the mini column below it's active.
Right.
Right. So that was confusing around me. So it's
Okay, yeah,
because over here, that's what I was suggesting.
yeah,
Right? This is where each cell up is like a grid cell, but it's a sparse SDR. This is more each mini column represents movement is active during a movement in a particular direction. So as you move in a particular direction, the active mini columns don't change. Yeah, all the changes that since the velocity of the cells, the frequency of which they're firing increases with speed, then which cell is in phase with the beta cycle changes. That's all it is. So
yeah, I guess, yeah. It feels like if this would actually work, if it does help, like, kind of, yeah, re-anchor the vector cells or like movement vector cells periodically, that would at least help with, because that always seemed like the main limitation of that approach was
what
was that you couldn't kind of path integrate over like slightly larger distances or like,
I'm confused by that. I don't know where you're going with that. That's out of the blue, it seems to me.
I thought that's what we were talking about just before. I mean, maybe you can ignore it then, but yeah.
No, what we're trying to, look, let's go back to the big picture. The big
Yeah.
picture is we have to have a sparse SDR, and we also have a less sparse SDR. And that represents location. And we proposed one mechanism for doing that. And that mechanism doesn't really have capacity for the less sparse one. It doesn't have enough capacity. So we hadn't resolved. That's the capacity of the morphological model. But now we're talking about the details of how you would get the more sparse model. And this is a way of getting the more sparse model that might fit better with the biology. So take the grid cells out of the picture right now. If you can just assume that at any point in time, you can anchor which cell is in phase with the beta cycle, then as you move, layer 4 would read that out. It would read it out as a sparse SDR, meaning it's the cells that are in phase right now with the beta cycle are sparse. So it solves that problem. It doesn't tell me at all how do I get the less sparse SDR for the morphology one. I don't have an answer to that right now.
As in, yeah, because you're still concerned that one layer of grid cells isn't,
Right.
although it's less sparse, it's not sparse enough. And then I guess that's.
One
what
layer
we're
grid cells is not very
that,
sparse
like,
at all. It's just like
if we
1 out of
only
30
have
or
a
one.
few morphological objects, then it's less of an issue,
Yeah,
like the.
yeah.
I'm debating whether I bring up another topics related to this.
Did you want to have a look at that Thompson paper about the
Yeah.
connectivity or?
Well,
Not that
I think you
relevant
brought it up, didn't it? It was this. You were going to argue that it was negative, it was inhibitory connectivity. Did she say that?
I'm not sure if that can, I think that's from the 2010 paper. Let me just search for that one. Oh, that is, okay, that's it.
There's a general rule about cortical connectivity, which is important here. But it's relevant here. Well, like the scroll up a little bit, I just saw some inhibitory connections.
So here it says in contrast to the relatively weak excitatory input from layer 4 to 6, there appears to be relatively strong inhibitory input.
All right, so totally understand, right? The layer 4 input is on distal dendrites or something like that. And so if you look at the soma and you see how much depolarization it has, it's like very little. So they say, oh, it's weak.
Yeah.
But they're not taking into account of the fact of integration in dendrites, right? You follow what I'm saying?
Yeah,
Right, so they'll say, oh, look, I measured the response, it's weak, it doesn't do anything. But that's what it looks like until you get a dendritic spike, and then it's not weak.
yeah.
Um,
And I forget, are they mostly based on measurements or on
I don't know.
anatomy? Because I think
well,
they also looked at the anatomy, like the
well,
connectivity.
why would you say relatively strong and
Yeah,
relatively
okay,
weak?
it's input, yeah.
Here, even then, here's a general rule of cortical connectivity. The general rule is the following. You have a cell, like a excitatory cell, and it's projecting to some other place. There's a bunch of cells here, there's a bunch of cells here, and it's going to make some connections here, right? The general rule is there will always be almost, not I would say always, but very often, this projects to an inhibitory cell here, and it projects to excitatory cells at the same time. And the inhibitory cell, these synapses tend to be right on the soma or next to soma. That's why you might say they're strong.
Yeah,
You're not recognizing a pattern. You're activating inhibition, global inhibition here.
yeah,
And so these guys, you want these guys to respond very quickly and on almost any input. So it's not like this is detecting a pattern. It's like, oh, I have immediate inhibitory response, where these will be on the distal dendrites and it'll look like a weak response.
yeah. That might match with this sentence here. So layer 4 double bouquet cells with an adaptive firing pattern bundle of fine descending unmyelated axons and more distal dendritic targets also innervates layer 6.
Well, let's read that again. That's really good. Double K cells with an adapting firing pattern, a bundle of fine descending unmyelated axons, right? And more, is that a sentence?
Yeah, the
How do I
sentence
parse the
seems a
sentence?
bit odd. It feels like there should be a comma somewhere.
Something, I'm
here, but
not parsing that
I,
sentence.
I guess what I'm, like, the more distal dendritic targets, like basically layer 4 double bouquet cells going towards the distal dendrites in layer 6 is how I read it.
Let me read it again. Layer 4 double K cells.
Oh, this is the verb. And then,
What's the verb?
I thought that's
No,
the
that's
adjective.
that's an
I think
adjective.
it's also innervate, right?
descending,
It's like,
oh,
Okay, the layer 4 cells with adapting firing pattern, abundant science axon, and more distal dendrites. Also, yeah, also innervate layer 6.
it's like
Um,
just describing them. Yeah, that is a weird sentence.
Okay,
Yeah.
well, you know what that sentence is saying? That these cells right
Yeah.
here,
Go to the distal dendrites.
they
in
go
layer
down to
6.
layer 6.
Yeah.
I'm not sure what cells in layer 6 they're talking about there, but they're saying I see an axon from those cells coming down to layer 6.
Yeah,
That's the exact data that I was saying you would want to see.
Yeah.
So,
yeah, that's
Yeah,
nice.
and also for what it's worth, you're talking about what is the delay in case
yeah,
that would be
unmyelinated, right?
unmyelinated and also fine, because the
Yeah,
narrower
right.
they are, the slower they are.
Right. So,
There might be a delay.
so those, I don't know what he says also, because I'm saying they need to activate these guys. And then it says they also activate these guys, right? So these things here, we want the slowness here, really, right? We want the slowness here because we want to, well, here, we want it here. All right, hang on.
Yeah, the only odd thing is that this is under inhibitory inputs to layer 6, so I'm not sure if this are inhibitory.
Well, okay, so these double bouquet
Yeah,
cells,
these inhibitory connections are reciprocal. Layer 5 interneurons projecting to layer 3, while layer 6 interneurons project to layer 4.
I have to read this again carefully. Here's another weird thing about these. I think the double bouquet cells sometimes act as excitatory cells and sometimes act as inhibitory cells. They have this dual
Interesting.
phase to them. I don't know how they do this.
What?
Yes, it's like, I forget, it was weird and there's some explanation for it, how they do this. It just stuck in my head that they do that
Okay.
somehow. Well, okay. I still want to just sort of get back to this issue of capacity. I think that's a problem.
Okay.
Let's say that these grid cells are not actually the ones that project. They aren't the code we're going to use for our morphology model. They just don't seem to have the information for a morphology model. Like, they're just too limited. What I need is something that's less dense than less sparse than the sparse one. I need like, I need a little bit more capacity. How do I phrase that right? Maybe these grid cells are acting as we were just talking about a moment ago. They're really acting as sort of this buffer to keep track of which cells should be active at any point in time, that kind of thing. I don't know. It's like I need a less dense. I mean, a less sparse and a sparse. I need a less sparse and a sparse with separate connectivity between the two. And I was hoping it would be these double bouquet cells. But they can't be just grid cells. Grid cells can't, in their classic form, don't have enough capacity.
Do we have to have the classic grid cells there?
Well, I just assume the grid cells exist because the whole idea is we didn't have anything, how the cortex could represent location in a reference frame. And grid cells seem to fit the bill, at least the grid cell mechanism as a whole seemed to fit the bill, so we just assumed it would be grid cells. And people see grid cells, so they're there, it seems to be.
But they see them I mean, it's only the fMRI study
Right,
with
but
abstract
Right,
concepts,
but
so
it's, well, no, they also, there was a Chinese lab that saw them in V1 and S1. And then other people dismissed it like, oh, I can't believe they're there. I remember there was, the fact that this Chinese lab sent me their paper, said, look, we found them in V1 and S1. And then when I talked to someone like, I forget who it was, the
We
guy,
definitely had a meeting with Tim Behrens, and
was he the one who said that he dismissed it?
yeah, I mean, I think he was arguing at least that they weren't kind of in the egocentric or like object-centric coordinates or something, but I don't know if they
Maybe like
actually
that.
tested
I
that.
mean, a few people I've talked to about it, they kind of, they're like, I can't be right, you know, it doesn't make sense or whatever. And I'm like, oh, come on, don't dismiss it, try to work it out, you know?
I think there were at least some concerns about the statistical
Maybe.
measures they use, but I don't know.
Right. Well, I believe they're gonna be there. They're gonna be everywhere.
yeah, I'm just saying if we can figure out with a different mechanism like these, what, like maybe just these 1D movement vectors. And
Yeah.
then I think the data that there is for grid cells in the brain we might be able to explain it in different ways, like it's just moving through conceptual
Mm-hmm.
space that they measured there.
You know, well, I don't know why you want to bring in conceptual space. That just confuses me.
Oh, I just mean like, if we find a solution that works without the classic grid cells, it's not like this experimental data.
Right, well, I do need a solution that has path integration of I have two different the current hypothesis, we have to have two different populations of location cells. One would be for morphology and one would be for feature-based models.
Just going back to that assumption, I'm just thinking, or like wanted to revisit, like how much of an issue is it if, I think, yeah, kind of to your point, Vivi, but like, okay, we have the grid cells, but let's assume those are just for stabilizing and like re-anchoring the
Yeah,
vector
right.
cells. If we just use those vector cells for both of those,
I mean down in the lower layers, like
yeah,
the phase.
that's ultimately like the location code or whatever that reactivates both double bouquet cells for morphological features and then L4 cells for features. But it's just like,
Well,
we'll in general, we might have more like morphological features will be dense, whereas features will be sparse or something, but like, it's ultimately the same like substrate there.
well, here's one way it could work.
I'm just thinking if that's actually an issue,
Is what's an issue.
just like reusing, like just thinking in terms of like Monty, like we have kind of a model and then at every location there's a morphological feature, but at some locations there's a feature, a non-morphological feature.
Well, so,
And then it would be like a separate thing whether
But-
we like fuse.
but wouldn't you need we're assuming there's a unique locations for unique objects. So, you know, an object
Yeah.
that's unique with the, you know. And then there has to be a non-unique location for the morphology. Or less unique. I mean, I don't, like, when I'm on the coffee cup, I have a coffee cup with a logo and without a logo. Two identical coffee cups.
Yeah,
Well, I want to be able to use the standard representations there's just be a common spatial representation for both of them. But a unique one that is only useful for the one with the logo on it.
yeah.
So I want to have two of these sort of,
Is the,
I don't want to have to, you know, I want all the cylinders to have this, all the coffee cups to have
Right.
the same.
is the like ID feedback
ID.
enough for that, or like from
No.
L3 and L2,
I
like?
don't think so. I think I need two. I need to have one be a sparse version of the other one. Wow. Hmm. Well,
Like, and again, in terms of like how we might implement it in Monty, it's like you kind of have the same graph, but when you have a strong hypothesis that you're on a particular object, there will be unique unique features that you will predict at a location that you wouldn't have predicted otherwise.
Well, then you have to have some
Yeah.
way of knowing. Okay, you have to some way of, here, I'm on locations on a cylinder. But I have to have some way of knowing that I have a unique location on the cylinder with the logo. And I just have to have both of those. I have to have both of those, it has to be unique, because another cylinder with a different logo or a different feature needs to have a different sparse SDR for location.
So like in Monty, it would basically be like having one morphological model of a cup, but then having multiple possible feature maps for that cup. So like, but for each feature map, we still need to have a mapping of where the feature would be on the morphological model. So how do the locations in
Well, they
the
have to
feature
track
space-
together, right? I mean,
Yeah, so when we are, let's say we know we are on a coffee mug, we're on the body of the coffee mug. And then we have multiple possible feature maps that could be on there, like the Meta logo and a different logo,
Yeah.
whatever. So in the feature map space, we need to have a mapping of which locations on the coffee cup morphology correspond to which location in the feature map space.
yeah, although the beauty of the doing this with SDRs is that I just create a sparse version of the first one and it
Yeah.
works.
Yeah, and I mean, in the Monty implementation, it would also be pretty straightforward to make the mapping or to make it work, but yeah.
Yeah, I'm just thinking in terms of,
So it's not. Okay, sorry.
yeah, how it would maybe work at a neural level. So Ramy, I feel like you're good about reminding about the top-down feedback. So I'm just thinking about how, like, once you have like an ID here, whatever the IDs are there, like you have a unique location for the general morphological reference frame,
Well,
like coffee cup,
Right, but
and then
that has to be shared against all coffee cups, right?
yeah. And so let's say you're at the location that is associated with the logo on one of the coffee cups. That's going to send some positive signal here, but only with a sufficient prediction. Maybe it gets kind of
You
circular.
like, sparsify it based on a particular object.
Yeah, I was just thinking like, if you know you're on that object, then together that's enough, and then those ones would, well, and assuming you get the input. Yeah, like, okay, so let's say the Numento logo it has two logos. So you saw the logo on one side, you go around to the other side. You know you're on this object.
Okay.
Because ultimately, like, it doesn't make sense to predict you're on the Numento logo unless you know you're on the Numento
Well,
logo.
I can, even if it's on one side, I can predict when I turn it, I'll see it, right? It's.
But until you know it's the Numento logo, we don't want these
Once
cells
I've
to
seen it
fire.
once and I rotate it, I still know I'm on the momentum.
Sure.
It's persistent.
Yeah, okay, it doesn't matter if, okay, but my point is, it's the Numenta logo.
It's the cup of the logo.
Yeah, and then, so you go to the location where the logo is expected. You're going to get the prediction of that feature, which is going to be biased by the ID. If you're on a different cup now, without the logo, then you have the same location active. That's because that location is associated with the Numento logo in one object's instance, that is going to send some bias up to L4, but in the absence of the bias from the ID representation, it's not going to be enough.
I think, I didn't follow it, but I think what you're trying to say is we're gonna have a single reference frame and the prediction's I'm gonna make the unique prediction based on the object ID.
Yeah.
I don't like that. I don't think it's gonna work. Maybe I'll have to try to convince you that it's not gonna work, but
Okay.
it doesn't seem like it's gonna work. It's
I thought that's what apical dendrites were all about.
well, wait,
No.
come on, that doesn't make sense. Wait, come on, that's a curveball throwing. What's April standards all about?
So how would we know that this is a unique location on that object without having the object ID?
You wouldn't.
I think the proposal is you'd have a totally different location representation for when it's the location on the Numenta mug versus location.
How
on
would it be
different
totally
object.
different? Because I need to do, continue to need to do path integration. While
No, it's the sparse code, the additional, isn't that what you were
But it would still be,
suggesting?
both of them would be a subset of the dense code, so they would be, I guess, similar in some regards.
is that, are you proposing a subset of dense code, or are you proposing that just
Like
the
in this-
ID tells you what to do somehow?
Oh, no, I'm not talking about the idea, I'm just
Oh, you were
saying
talking about
that,
that.
Yeah, but I think Rami was asking, like, what is the alternative? And I was trying to paraphrase what we had been talking about before, which was that we have the dense grid cell code, the sparse, say, grid cell code. And it's the sparse grid cell code, which will have a unique location only for the Numenta logo at that.
When you're not there, what's the location representation? How do we go from a dense to a sparse one? Where's the sparse one and where's the dense one?
I thought that was the two populations we were talking about before. In L6.
Yeah, but before I had it as the grid cells, and
Yeah, that's what I'm talking about.
well, grid cells aren't sparse enough. They're not dense, they're too dense. They're not gonna work for us, right? I'm confused.
I thought we
Yeah,
got
I was just
this.
trying to paraphrase the previous argument. I wasn't saying that that was right. I was just trying to say to Rami, like, because we were talking about how are we gonna capture this? I was just talking about the ID, and then Rami was saying, well, like, if we're not using the ID, what are we using? And then I was saying, well,
Oh.
one thing we were discussing before was the two populations of
Right.
grid cells.
Okay, all right, oh, right, okay, that's what you're saying, then we're back to where we were before.
Yeah.
So we don't have an ID, we have a union and that in the sparse grid cells.
I guess until we know which object it is. Like, yeah, if there were two cups that both had logos in the same position. And we don't know which mug it is yet,
And then there's.
I think you would have a union of the locations.
And now
that
we
were
have to
associated
have
with.
integrating on multiple.
Yeah,
My summary of this is, what if we're going for this idea down here, where the mini columns are just phase distributions, then we have a solution that works for the sparse code. It works. I can encode that in layer 4, recognize that pattern. I'd have to recognize different patterns going in different directions. I'd have to say, going this direction is, if I approach a point this way, it's gonna be a different pattern code this way, but it could work. I don't have now the code for morphology. I need a less sparse one for morphology that follows the same rules. 'Cause the first thing I said, we have to have two models that share path integration. And so that's the bottom line here. We have to have two models, one for morphology and one for specific ones that follow both through path integration. And the morphology model is active in different unique objects.
Did I miss something? Oh, what's this proposal now? I thought this was that there are still grid cells in the,
Right, but we're not using the
Fine.
grid cells, we're not using the grid cells for, the idea is that grid cells are there as a maintenance function for the other cells. They're not actually encoding anything. They just don't have enough information to encode anything.
so we couldn't use those two.
Well,
steps.
if we tried to, then we have a capacity issue,
Yeah,
right? The capacity issue is that if you literally take the tank paper and you say, oh, well, really, I just got, how many different locations can I encode in a grid cell module? I don't know, 150, whatever, that's. It doesn't work, I mean,
I'm just thinking if the top-down feedback with the ID thing, if that helped as well in terms of, yeah, path integrating over multiple objects. Cause that was like another issue we discussed yesterday, is like, how do you test multiple hypotheses at once? But basically, if we assume that there's just like one, like, it's not separated into morphology and feature in terms of the location representation, like for mugs, we just have a common one. Then naturally, when we move through that, we're gonna be kind of testing all hypotheses, because the location isn't unique.
by the way,
But then what's unique is the activation that will happen in L4 when you get the ID bias.
the ID's got a problem too. Because we have to have two separate models. I have to be able to have an ID for the morphology model. I have to have an ID for the feature model. I can say that's a circle, right? if I need to be able to identify that and pass it on to someone else. So,
But that feels like maybe a separate issue for like,
I don't know, it feels like I need to have two, I have the same issue here, but it's like I have to have an ID for the common object, I have to have an ID for the unique object. A convenient way of doing that would be to sparsify the ID for the common object.
Should we maybe just collect the main issues right now and then take a little break?
Sure.
Just add to the list.
yeah,
Well, I don't know, I actually don't think we're too far away from having an answer, I think it's pretty close. actually.
Yeah, I think it,
Uh,
there's some really good ideas in here.
I mean, the best summary I can do is what I just said a moment ago. You have to have two models that track together from path integration. And it seems like the surest way of doing that is to have a sparser version of the morphology model has a set of representations for features at locations, and you want to sparsify the locations and you want to sparsify the features. That would be the way, the simplest way of doing the two models. We have, I mean, we also agree the capacity of the morphology model, we have to have more of the, I'll make a statement, we have to have more of the feature models than we have to have of the capacity models, because the feature models, many of them share the same, I mean, the morphology models, so many of the feature models share the same morphology. Therefore, we have to have higher capacity in the feature models than in the morphology model.
but they also might be more sparse, as in like more. They encode less information in that each feature model is probably a few points of information that need encoding. Whereas
Right.
the morphological model needs to kind of densely encode.
Right,
the
I guess
whole-
that's true,
The whole
um,
morphology.
right,
But
I guess,
even the feature models might need to be dense, like you wanna have colors at all locations in the.
But like that would, I'm just thinking that would be like. Where you could. It feels intuitive, like you could easily learn like the detailed morphology of an object, but to like learn the color at every location, if it was like, let's say some crazy pattern. Like you probably wouldn't memorize that,
Yeah,
but I don't know.
uh,
Or like if a cup was covered in like hundreds of like logos. You'd probably just
You wouldn't-
assign
you
some
would-
sort of like generic.
Right, you wouldn't say,
Uh,
here's what
you
this
wouldn't
logo is.
memorize each
Yeah, I mean, it's an
feature.
interesting question, we haven't really addressed that question, like, oh, if the whole cup is red, I don't have to learn red at every location, I just somehow.
Yeah,
Maybe you do, because the different columns are looking at different points and they're all learning it's red, I don't know. Well, I think the new idea today, we'll revisit it, but new idea today is going back to this idea on the bottom left here as a way of getting the sparse coding of locations. And only reason I like it is because that's what they observe. That is known to be observed in Cortex, right? You got mini columns of orientation, movement, preference, orientation in lower layers. So we know cells that look like that are there. No one's reported that they're phase shifted, but that would be a prediction of the model that the cells in the mini columns be phase shifted. What we don't have is a denser model. We don't have a good working model of the denser model yet.
and. But at time, yeah, I'd like to kind of just try and think through maybe later, like more about ways that we could reuse the same substrate. Like I appreciate there might be issues with it, but at least that is one solution, if we can find a way to it, of like, instead of having two separate populations, if we can somehow reuse the same population
Right.
for the reference frame, then we don't have the population, a problem from like a neuro biological.
Well, but you do want separate connections. You want to be able to say, I need to make a unique prediction. I think they still have to be separate. I mean, they can be the same substrate, but they have to. It seems like the models have to be somewhat separated. I don't know. Can so we're gonna take a break now? Can I throw out one last thing before we take a break?
Yeah.
Sure.
This may be a total red herring. But in vision, in a V1 column, what you see is now I'm looking at, remember, this has to do with the slabs. Remember the slabs? You see
What
those?
slabs?
Here's what in V1, if you move your probe in one direction across the quarter, what you see is, you see the mini columns change in orientation. Like that, right?
Mhm.
So in this direction, you see that. And they cycle through again and again. If you go in the other direction, no, didn't do that right. You go in the other direction like this. The mini columns all have the same orientation. And so why? Why is it that these all mini columns all seem to have the same orientation? They don't seem to be different from each other. Whereas you can go in this direction, they're all different from each other.
But so you're talking about something different from the penetration.
I'm just saying, if you look on the surface and you say, okay, the mini columns, all the cells in the mini column here, this is the mini column. And they stick to penetration like this, all these cells respond to the same
Right.
orientation. Then you go to this mini column, all the cells respond to the same orientation. So as you move in one direction across the cortex,
Yeah.
the orientation changes continuously. You move in the other direction across the cortex, the orientation doesn't change at all.
Is it, I thought there were like these pictures with like the pinwheel color.
There is, right, so this is a well understood phenomenon, but then you overlay the pinwheel on top of it, okay?
Oh,
You just, if you look at the Hubble and Wiesel iceberg, ice cube model, you just go to Google Images and you'll see it. Right now, I can do it. Google Images. Let's see, yeah, maybe that one there. Right there, okay?
maybe.
So right here, this picture right here. See, these, if you go in this direction, the orientation's changing.
And then it's the
Like when you go
ocular,
across this, right here,
it's the ocular dominance.
well, this is left eye, right eye, but within this little rectangular block here, there's multiple mini columns, all have the same orientation.
Oh, yeah, yeah.
So like you might even see it better here, perhaps.
And then you still have the blobs and interblobs.
Yeah, I
Okay,
didn't want to go, take this one up here. Oh,
Oh, it's a very small
I didn't
picture.
get it, well, maybe it doesn't show up there. Anyway, you can look up, I'm pretty sure of this, I mean, I'm sure of it, is the way it looks like. You have they call it a slab because a slab of mini columns, not many, but a slab of mini columns, all the same orientation.
Mhm.
And the next slab over has a different orientation, right? And I've always wondered about this. It's like, what the hell's going on there? There's some redundancy there, but I don't think it's redundancy. It's some part of representational scheme. I don't know if this applies to anywhere else. This might have to do purely with three-dimensional image viewing, like trying to get perspectives. There's a whole bunch of things that could have to do with absolutely nothing to do with our problem here.
In this picture, it looks like it might be a spatial frequency that's encoded along that direction.
I don't know. Is that what it is?
I don't know, maybe they just suggest
Oh,
that,
I didn't know
but,
that. When was this?
modified ice cube model of cat V1
Hypercolumn.
column. But I don't know,
It could be.
I can share that paper if that.
I don't know. What
Um,
the hell is it? What does that
so
mean?
you have like, there they show that direction and the ocular dominance columns, but then within one ocular dominance column,
Oh,
they seem to
okay.
assign spatial
I
frequencies.
I guess
to those.
if
maybe
it was
it-
like sinusoidal gratings,
Why
it
would
would
they,
be like
why would you do
coarse
that?
ones versus fine grain.
Right, why would you want that? Okay, I said it could be a total red herring, it may be a total red herring. It's maybe something specific to vision, because vision we have the 3D, perception of depth, we have, viewing at a distance, you have to calculate the distance of things, there's a whole bunch of stuff going on in vision that doesn't occur anywhere else. So it certainly doesn't occur in the somatic sensory, so maybe we shouldn't pay attention. I was kind of come up with other ways that the reason I thought of it, here's the reason I thought of it. I said, oh, well, I
I could use this other dimension here to create a different way of creating unique morphological models. It's like it gives me two variables. It gives me, which cell is active in the minicolumn, but also I can tell which of the minicolumns are, something about this dimension. I was just trying to think of other ways of increasing the capacity or changing the capacity of the representation. It's probably a red herring. I probably shouldn't have brought it up.
Yeah.
Okay, we're gonna take a break.